Well, welcome to today's Safer World by Design webinar. We have an exciting topic today on multi-organ chips. They're increasingly finding applications in different kinds of applications that can be used in safety and efficacy testing and supporting our ambitious goals towards next generation risk assessment, like we're working on together. And happy to have Beren Atac Wagegg with us from TissUse. And she's going to take you through the presentation, which will take about 30 minutes. Just remind you there's a question box there to submit your questions, and look forward to a good discussion at the end. So with that, over to you, Beren. Looking forward to the talk.
Thank you, Barry, and hello everyone who is joining. And I'm happy to tell you a little bit of the story, what we have done, and where we want to go. It's with some examples what we have done so far. So I want to basically start with a little introduction of who we are. We are a spin-off company from Technical University of Berlin that we are founded in 2010. We are still a small company, at the moment about 50 people, and we have a connections, a strong customer base in industry and academia. And our technology is patent-based, that we have many patent families in place. And since actually 2019, we have a quality management system in place. And this is also bringing me to what we do. We are trying to find a solution or improve the problem, or provide some solutions to the drug testing dilemma, mainly that there's a very high failure rate in the preclinical to clinical stage, that most of the new compound entities are actually falling off the stages of the clinical trials, and we see the problem that that there can be improvements done at the preclinical stage and the first phases of the trials. I mean, this is from the drug testing point of view, but this could also be applied to the toxicology point of view. In the end, what we are trying to do is taking the best of systemic testing, which is done on animals, and human testing, which is mostly done in 2D and 3D simple cell culture together, and to make it both human and systemic, to have more relevant test systems in place. So in this case, this is really far ahead. What we want to have is a universal physiological template, like a human on a chip, so to say, to basically test the patient, have personalized results for whatever we are testing, considering the genetic background into account for both safety and efficacy. And this could, and we really strongly believe that this could improve the compound testing and also finding new drugs and bringing them to the market in a much more successful rate. So what does it look like? Let's say how the multi-organ chip system looks like is just to give you a bit of a feeling. These are chips in the size of a credit card. It could be 2-organ, 3-organ, 4-organ combinations. We're gonna see some more examples later. And basically, this is, let's say, the hardware, and then we use either different type of biological parts. This could be cells derived from iPSCs. These could be cell lines, primary cells. It could be biopsies like ex vivo tissue. it could be tissue slices that we have. These are the things we have tried so far. And here you see some examples of the type of organ models we have tried in the chip so far. So idea is to have a relevant organ combination that would be a fitting assay. It's a fit for purpose assay that would be coming together in the relevant chip, depending on what we want to do. So it's a very flexible system in that sense. And if you look a little bit more closer how the chip looks like, we are looking into the 2-organ chip in close, and it's a transparent system, so it's microscopable. There's a glass slide under it. Here we see the example of PDMS with the microfluidic part on it. I guess you can see my cursor. It can be also, we also work with other, actually other materials than PDMS, but most of the time PDMS. So these are basically the organ compartments where we can place the tissues, and it can either be directly put in, it could be with transwells, for example. And then we characterize also the system, how the flow characteristics are, especially when we develop a new chip system, for example. And for us also, the very important part from the very beginning to solve is that to create a pulsatile flow that actually creates a flow that is a heart-like. Also comparable in terms of the speed, in terms of the pressure, like the blood pressure, and it's a closed system like we do, like we have. So once these are like the insert lids. So once the lids are closed, it's a closed circuit that the medium just circulates around. And these are where the pump actually controls the flow, and there is like the pump actually does not pump any medium in and out since it's a closed circuit. It just is controlling little membranes over this area, and these membranes are basically moving up and down, creating the flow. So this is the basic structure of the chip we have, and having let's say the technology in mind, I want to direct you a little bit to the what we expect from the microphysiological systems that would come to the patient's benefit, because yes, we like the technology is there, the biological models are there, but which steps do we need to take? So obviously we need to have qualified equipment and devices that we know that how it behaves, how reproducible it is, how reliable it is. And then it has to be, then we have to basically bring it together with biological models. I mean, in the beginning, it starts with academical basic research models. These biological models we need to characterize, we need to understand how it works. And then once it comes together, it is a good biological model. We know what to expect. We know the limitations, we know the strengths. Then basically we need to so the third level is to make it into a qualified assay. This assay that we know, okay, this with these organs together, we are aiming to test this, and then we are aiming to have this outcome. And again, this is something we need to understand the strengths and the weaknesses of the predictability of these assays. So there are a lot of steps to achieve, and eventually where we need to come is a validated assay that is recognized by the regulators. And so with this, basically the next step, or where we are right now, is that we have different level of assays where we establish just some examples, like in this case, like the first stage is a proof of concept assays we have done. We have established that they, that these are the ones we are pushing forward for an established model to have this assay developed further. But we already have 7 assays that are done with the proof of concept. So as an established assay that is already in regular use. And from this point of view, we don't have any validated assays. And this point of view, the next step, so it's actually a big step that we are trying to also understand, and we need to understand how we can standardize, how we can go further to basically provide the benefit to the patient, that everything is that the timeline of the clinical trials are maybe shorter eventually. I also now want to show you some of the research we have done, and from that today I try to organize them a bit more liver centric assays, mainly that also has liver in. So these are just some examples that we did with liver so far, and now from this part, we can maybe start with the skin and tumor culture, which is actually not only with not with liver. This is also the one that is without liver, to mark that you're not you're not wondering. The interesting part of this project was that we looked into both safety and efficacy aspect of the at the same time, or in this setting, in this assay. What we tested was lung tumor cells, and in co-culture with full thickness skin equivalent. And here we applied an antibody. This is EGF receptor blocking antibody. This is a usual, chemotherapeutic agent that is used in the clinic. So it's a proof of concept study. We know what to expect. And this a very common side effect here of this chemotherapy treatment is that since it's an EGF receptor antagonist, it also actually has a really large side effect on the skin. And what we wanted to replicate was that, okay, do we see the treated tumor recessing? Do we see the apoptosis in the tumor? Do we see the skin being affected? And indeed, we saw cell death in the tumor, and we saw a diminishing proliferating cells in the skin. And this is also was for us being able to show, yes, we could replicate the skin toxicity. We could show the efficacy on the tumor. And from skin point of view, now I'm coming to the liver part. We did in a different project, we did toxicokinetics and toxicodynamics in this combination, and we basically compared in this setting routes. So it's either topical or it is systemic application versus with a single dose application or repeated dose application. So this was to compare also how the metabolite profile would be. So here, this was the retinoic acid. Again, we mainly use the compounds we know what we are expecting to see. The nice thing, for example, we showed in the single versus repeated dose testing for the retinoic acid on the topical part was the one of the metabolites were only coming after 48 hours, which was basically after repeated treatment. And in a single dose treatment, we did not see this metabolite coming up. In the systemic treatments, we see all the metabolites coming up earlier, and you see also the metabolite profile and the amounts that are produced are actually different depending on the route. And we also then looked into from this is permethrin, another in the context of, again, topical versus systemic, single versus repeated. And the NICE actually also in towards reproducibility and interlaboratory technology transfer wise, we also did all the same experiments in 2 locations. So this was indeed we could show comparable results. This was the systemic application, and I believe this is also something very important to be able to eventually qualify any assay that should be done multicentral. Next point is the towards establishing a thyroid liver assay, and this was aiming to identify towards basically establishing an endocrine disruptor assay. For that, we first need to understand obviously the models, the liver, the thyroid model that we have is actually is expected, working as expected. So in this case, the liver component is important because of the metabolism of the thyroid hormones that is expected to take place in the liver. So in this co-culture system, we characterized first the gene expression that in the liver and both in the thyroid models. Do they have the thyroid carrier protein expression? Is also the hormone secretion? Do we have also the hormone secretion in the thyroid as we actually expect to see? And the nice part of this project was that we did everything in parallel in the rat system and in the human system. In an important aspect is that since we have many data on animals, we also at some point need to have the animal comparison in the chip to be able to say, yes, we can also replicate the data at the proof of concept level, that we can also replicate the animal data. So what was very actually interesting to see was that in one of the metabolites, the sulfonated T4, we could detect in the human setting, in the human liver models that, but this was not the case for the rat. And so this is also another indication that when we are using a rat system, that for hormone disruptors, hormone disruptor testing, we may not, so we have to take these differences into account. And next one is then with liver and pancreas, and you can imagine what we looked into. We wanted to look into the diabetes model, so to basically to look into the crosstalk of liver and pancreas. And here we first needed to set the model and look into, do they really talk with each other? And indeed, we used pancreatic islets and liver spheroids. This was basically HepRG based, and we did their co-culture for 15 days. In between, did glucose tolerance test 3 times. This is for 48 hours. And we looked into the responses. So what we had to show or wanted to show was that in the presence of liver, the insulin secretion, for example, here is regulated, and vice versa. In the presence of islets and pancreatic islets, the glucose uptake is taking place by the liver. And when we are culturing only liver, these are either islets only or liver only, we did not see a change in the glucose levels too much. So in the case of insulin secretion, we could also show when the islets are there in the culture alone, that their insulin secretion is much more than when they are in co-culture. So that this is a known effect, that the liver tells the pancreas, okay, so it's a negative feedback loop. We have enough, so this is enough insulin, and that pancreas do not have to secrete constantly high amount of insulin. So this also has an end effect. So in the longer term, when in the co-culture of liver and islets, the functionality of islets were kept longer in comparison to islets only, since the islets were cultured alone and their insulin levels are constantly high, eventually the levels actually decrease. And in the liver and islets were cultured together, basically the secretion is more or less stable over the time. So in the case of it, or in the direction to bring it to the diabetes, so this is basically until this level we were establishing the crosstalk. And in the next stage, we wanted to include a computational approach into this. And here the research involved, okay, again, it's the liver pancreas co-culture, and the computational model has been first. The model is developed, and then it has been calibrated, and then eventually challenged with and evaluated with the data we collect. And here, what has been shown is that that glucose concentration and the liver islet co-culture, so it has been challenged in with normoxia and with the hyperglycemia, so or normoglycemia, I'm sorry, normoglycemia and hyperglycemia. And these values have been used for calibration. This was 7 experiments, and in the end, hypoglycemia was used for prediction, or hypoglycemia was predicted by the model. I'm sorry that I forgot to add the reference here. I can share that later. And the interesting, also what I really found interesting, was also the insulin concentration in this section was same, like again with the predictions. Here, these are the data values. You see the measured values, and these are from an independent experiment that was not used for calibration. So there are like 7 experiments used for calibration, and then there's another experiment that is used to see if there's basically an independent experiment is also if overlapping with this. Okay, so and then this is an in vitro model with this use the computational model. But the next question was, can we translate it to an in vivo response in humans? And here in the part glucose concentration in the liver compartment, glucose concentration in pancreas compartment, or versus insulin. These are the parts that has been predicted by the model and then compared to a human situation. These are the dots are the measured human values basically. And this also brings us together with the that we need to take into account modeling aspects, in silico approaches, together with the in vitro data to come closer to what we can predict better with the in vitro test systems. Okay, speaking of modeling and complex systems, here basically I come to our most complex assay yet, and this is a ADME platform, that meaning it's absorption, distribution, metabolism, excretion, together with toxicity, with the toxicity target. And here basically the motivation is starting from the in vitro data to be able to predict an in vivo effect. Here, we need to do some measurements, obviously, and then modeling has to come into play, and then we need to come to the prediction part. For that, this was in the Autox Risk project, we designed a 4-organ chip. And here we took into account the physiological arrangement of the organs and with their physiological perfusion rates. And it looks basically, the chip looks like this from the side, and it has 2 circulations. One with the urine circulation, one with the blood circulation. And in the interface here, you see the interface, like there's a porous membrane for the kidney cells to basically be on this chip, on the membrane to create the kidney barrier. And when coming to the intestinal barrier, we have them in an insert, and this allows us basically to test for oral route. To mimic the oral route, we have a let's say medium reservoir to bring a compound on a systemic route. We have a liver compartment, and our toxicity target, especially for this project, was neuronal cells. And we have a neuronal compartment here. And in the kidney, we have proximal tubuli and glomerular compartment separately. So what we have done, so we are looking from top now. What we have done so far is that we cultured these all 4 tissues for 14 days together, and we use basically 2 administration ways, either the over intestine or also systemic, and then we looked for this distribution and effects on the neuronal part, and looked also if there's excretion as well. So, and over these time, we also detected that the kidney kept integrity, and also we could even see an effect due to the treatment. So we did use 2 known compounds to test to challenge the system, and but yeah, we will hopefully come to publish that soon. So I cannot disclose the results yet, but it looks interesting so far. Anyhow, so keeping these complex systems in mind, I want to bring to some discussion points, and just to stir up your thoughts a little bit. If you're using these complex systems, I mean, you starting from the toxicity or let's say test batteries the simpler systems are the ones used the most, and they are basically in this case qualified. And then when we go more complex and to emulate the human biology, we are also more far from the qualifications, so, or qualified systems. So how do we connect this gap? In the end, like I discussed in the beginning, I believe we need to understand the models we have, and it has to, let's say, fulfill certain expectations, certain biological responses that we need from this particular assay, particular aspect we are looking for. And then the test system needs to be refined, needs to be calibrated, and it should be reproducible, it should be robust in terms of also cost. It should be in the end more cost effective than all the other tests we are, that we are spending long term. And coming to the to confirm that it is really fit for purpose, we need to show the in vivo relevance, that it is really that we can really translate it. So this is basically the thoughts, the way I believe will step by step, with many other steps in between probably. And really looking really further in a bit more in the future, this is more in the drug discovery point, that I believe we are working on to have it to a level that, let's say, having the patient on the chip, to be able to have them to have it in parallel with the phase 1, phase 2 trials, and even having the healthy, like untreated healthy patients, or even having the healthy patient on the chip diseased, and then treating them in parallel. So having really clinical trials in parallel to the chip, until we could say, yes, we have some preliminary data to say, okay, we can really proceed, or we have more concrete data that we can proceed to phase 3 trials, for example. So this is where I stop, and then would like to answer some of your questions if I can.